Hello, I'm Sofie Valk, and I will speak about structural covariance from layer thickness to transdiagnostic impact. So thank you for seeing this video. I have no disclosures. So structural covariance, what is not to love? As I had also this afternoon about practicing this talk with my lab, I really have to say structural covariance is one of the most amazing metrics that one has of the brain. In particular, I will focus on cortical thickness covariance in this talk, but there's many different things you could covary in theory. In theory, you could covary everything. But I think for the focus of this workshop, focusing on cortical thickness covariance is the most informative because there I have done 3 studies together with also my lab. And what is so interesting about cortical thickness covariance is that it reflects shared maturational and genetic processes, and it's organized into multiple communities, as you see here, which has already been shown in 2009 by Schmidt et al. And here they did a genetic correlation of different regions, and they showed that they're clustered into different regions. And here with this hierarchical dendrogram, they see that some regions cluster together more than others. Because if I forgot to say, like, covariance is a measure across participants. So you look whether the thickness of a given seed, for example, here shows a positive correlation with the variation of thickness in other regions of the brain. And sometimes it does. So in this case, it would point to shared maturational, but also shared genetic factors overall. And sometimes it doesn't. And this would maybe mean that there is less similarity in maturational trajectory or even less genetic similarity between these 2 regions. So the first question I try to answer in my work is, what is then the genetic basis of the spatial organization of macroscope cortical features? Because in the past, it has been shown that it's organized in different clusters, but how then these clusters relate to each other was not exactly clear. And then also what this means was not so clear. So here I used the twin model enabled by the Human Connectome Project S1200 sample with monozygotic and dizygotic twins. And I also looked at a comparative model using the prime.de data set and a comparative model between humans and macaques. So first I found 2 axes because I made this covariance matrix and then looked at the genetic correlation of this covariance matrix using the twin model. And I did dimension reduction techniques to uncover its organization. So on the one hand, there was an axis going from more posterior regions here in blue to anterior regions here in yellow, showing that the regions with the same color would have a similar genetic or a higher genetic correlation with its own color than with regions in another color, making these regions in blue and yellow the most distinct genetic correlation. In macaques, I found a similar pattern, also going between unimodal regions of visual and sensory motor areas towards the more yellow regions, more in the anterior part of the brain overall. So this axis actually represents a hierarchy or a differentiation between sensory motor regions versus more transmodal areas in association cortices. On the other hand, I also observed a second pattern going more from inferior to superior parts of the brain in terms of genetic correlation of regions and how they relate to each other. And this pattern was also observed in macaques. This model then again related to the dual origin model, which is a conceptualization of the development of the cortex from 2 different origins, the piriform cortex, that is part of the amygdala, and on the other hand, the hippocampal area, as you see here. So in these ways of laminarization shape then ultimately the cortical mantle and its expansion across different mammals. So it seems, or what this study could show, is that there's 2 axes of genetic organization in the cortex uncoverable by studying the structural covariance and the genetic correlation thereof using the twin model. And in this way, it may provide a compass in organizing the structural communities that we observe in the brain. However, then together with Amin Sabiri, who is a PhD in my lab, we wanted to dig deeper, and we looked at the organization of laminar thickness covariance in the human cortex. So here you see the different laminae of the brain, which were annotated by Conrad, who was also part of the Symposium, Conrad Wachstil. And And you see that there's also subtle variation of the thickness across the different areas. So what we were thinking is that maybe this relative distribution of layer thickness also co-varies across the cortex, so that certain regions have a more similar laminar profile based on its thickness than others. So yeah, as you may well know, the layer of the cortex is mainly based on a 6-layered isocortex of different cell densities and also different cellular distributions. It has been shown indeed that there is variation in the cortex in terms of its laminar profiles. However, this is often theory driven and has a low sampling rate. And here we wanted to look at the whole brain and how it's variable. And for this, we use data-driven approaches, which I will explain to you now. So firstly, we used the big brain and a map of cortical layers using a 65-year-old male post-mortem brain. Cell bodies were stained, and it was reconstructed using ultra-high resolution 3D atlas in 20 micrometers. Then there was a layer segmentation using CNN, which is a machine learning approach to annotate different profiles, in this case here in the cortex, into layers. And we projected the volumetric mask of this towards the cortex, which was also done in a previous work by Conrad Wagstiel. Then we performed pre-processing, where we took these layer surface profiles. We excluded the A and the disgranular regions because here the layer differentiation is less clear, so it wouldn't make so much sense to look at the layer covariance. We did some smoothing to account for other effects of subtle variations that were just due to mismatches. And then we had the Bock effect, namely that there's a differences in thickness between the sulci and the gyri, and we wanted to correct for this. And this, the smoothing largely did, as you see here also, it's varied a little bit, like this and this. And then we normalized it by total thickness and applied the paracellation. And then we generated the relative laminar thickness in each region, as you see here. And you see there's quite a variation here across the cortex, but also potentially some similar profiles. So with this data, we performed a partial correlation, generating a laminar thickness covariance matrix. And with that, we did a principal component analysis to find the main gradient of laminar thickness covariations, which shows this more rostral caudal pattern between blue and yellow, as you see here. And if you order the laminar thickness profiles across this axis, you will find this beautiful organization here. And what is cool is that this differentiation actually differentiates layer 3 or supragranular layer thickness from those in the deeper layers, as you see here. So that's the pattern, or that is the pattern of laminar thickness that is actually underlying the dominant pattern that we observe here. What is also super nice, or at least for me as a structural covariance fan, is that we find that this layer thickness covariance relates to structural covariance. So this within subject analysis related to this group level analysis I presented in the first study. Indeed, we also then went further and we wanted to understand cell density and size and how it relates to our layer thickness maps. And for this, there were different sample locations, as you see here in this big brain, which enabled us to understand, like, would this then also show some other variations in morphometry. And we also related this then to average laminar density and combined maps of laminar thickness and density covariance. And what you see is that the map highly resembled the original laminar thickness gradient, but you see also that there's this variation of laminar thickness and density along these principal axis bins of our gradient. And what's more, you see that there's a positive relationship with laminar density, in particular in the layer 4 and 5. And again, an association with laminar thickness, as we've seen before. And what's more, looking more at the cell properties, we found that there's a relationship also with cell density, but also cell size, which is anti-correlated. So there's a negative relationship with cell density, as you see here, particularly in layer 4, but also layer 3 and 6, and a positive relationship in layer 3, 4, 5, and 6 for cell size. What's more, even we looked at external meatization, sorry, I cannot pronounce what we've done, but I know what we've done. Namely, we looked at the neuronal size of layer 3 and layer 5. So we looked at the ratio and then related this to the fused gradient loadings that we observed. And we found indeed a positive correlation with this metric. Then we also wanted to understand, like, is there maybe a functional relevance of this laminar structural variation? Because we know from the literature that differences in laminar structure relates to laminar patterns and the direction of connections. And this may then again relate to cortical functional processing. So for this, we used two models. On the one hand, we used a symmetry-based functional hierarchy using resting state data and weighted in and out degrees, and we compared them to each other. And we generated this map, as you showed here, and we then correlated this with our gradient, and indeed we found a negative correlation between a symmetry-based hierarchy and the first gradient of laminar thickness profiles. What's more, we then validated this also with track tracing data in macaques by projecting our gradient onto a macaque gradient and looking at laminar-based hierarchy based on the pattern of feedback and feed-forward connections. And indeed, then again, we see this relationship similar to the in vivo model, implying that maybe there's also a relationship in this feed-forward, feed-backward, or functional processing organization between these laminar profiles on the one hand and these two models on the other hand. So an interim summary would be is that we found a link between group level macroscale cortical thickness covariance and individual different layer thickness covariance. Layer thickness also differentiated layer and cellular features, and we found a spatial association with functional processing hierarchies using two different models. Last, I want to speak a bit about neuropsychiatric disorders and conditions, because we wondered, together with Maaike Hetwer, who's also a PhD in the lab, could covariance provide valuable information on the co-occurrence of structural alterations? So maybe there's some translational potential, because transdiagnostic co-alterations in neuropsychiatric and neurodevelopmental conditions happen because there's some genetic relationship between different neuropsychiatric conditions, such as anxiety disorders and major depressive disorders. And we also know that there's overlapping endophenotypes when you look at the impact they have on brain structure. And we also know that there's network-based pathology, implying that there's some coordinated changes in the brain, looking not at regional level, but inter-regional relationships. And last, using consortia and open science, we are able to use more normative map of certain disorders to compare like different clinical phenomena or maps to each other. So in this case, we use the Enigma maps of bipolar disorder, ADHD, autism spectrum, depression, obsessive-compulsive disorder, and schizophrenia. So this is all the Cohen's D-score of these disorders versus healthy controls in each of these conditions. And you see that blue indicates a negative, so less cortical thickness, and red indicates more cortical thickness in the respective condition relative to controls. And then the idea was, what if this impact of disorder also has a systematic co-alteration across disorders? So we created this transdiagnostic covariance matrix. And then we wanted to know what are then the main patterns that organize this transdiagnostic covariance. And we performed a dimension reduction technique to see what regions covary together across disorder. So I hope you still follow me, because what is interesting is that we find that regions with similar colors are affected together across disorders. So the first gradient, which was the main explainer of the variance across disorder, differentiated more temporal but also posterior regions from frontal regions, whereas the second gradient you see here is more posterior versus motor and the superior temporal areas. Indeed, again, we found a link to structural covariance, which I think is super interesting because it means that regions that are affected in the system level across disorder also link to this group level covariation, which again links to shared maturational and genetic effects, but also maybe even to these laminar profiles that I discussed earlier. And what is more is that these different dimensions of shared susceptibility may link also to different symptom dimensions that are observed across disorders and also shared across disorders in different ways. So on the one hand, we found a cluster using meta-analytical maps of neurosynths of sensory impact. A second cluster, what we observed in this covariation, was more an attention perceptual cluster. And a third cluster that we observed was related to cognitive control. So maybe these functions are also impacted in different ways in different hierarchies or organizational features within how structure is organized and co-organized in the brain. So our findings suggest independent maturational causes and distinct vulnerability at different functional processing levels observed in behavior. So to sum up, I can really recommend to chat with Maaike and Amin if you're interested in this work. And we learned a lot about covariance, and yeah, as to repeat, I think it's the best measure. So on the one hand, we learned how covariance reflects the sensory-transmodal differentiation, but may also put forward more ontogenetic patterning related to dual origin patterning. Second, we show that the group level covariance could actually link to layer-based covariance at the single subject level, differentiating supragranular and infragranular layers, but also functional processing hierarchies. And last but not least, covariance can also be observed when you look at a transdiagnostic model across disorder, which may help to understand how different symptom clusters relate to each other and co-occur along different disorder dimensions in different ways. So I hope this talk was interesting. Yeah, I want to thank my team and my collaborators. And yeah, by the way, I'm recruiting at all levels, so give me an email or talk to me, and happy to chat. So thank you so much. Goodbye.